it was that there was very limited information about the cells and tissue types that we are particularly interested in. So you've probably all heard of the PsycEncode project. This is really a follow-on to both of these. It's, again, a big consortium of people interested in mapping different layers of gene regulation across the genome, but this time specifically in the cells and tissue types related to neuropsychiatric disease, and also then relating these back to individuals who have disease and comparing or identifying signatures that seem to be associated with bipolar disorder, schizophrenia, and autism. So why are we interested in the epigenome? Well, your DNA sequence is obviously something that's very stable. Other than rare somatic mutations, the DNA sequence at vast majority of positions is highly stable and is identical across all the cells in your body from the moment of conception onwards. But of course, gene regulation has to be highly dynamic. And we know that there are changes that take place during development, as I've inferred. But the epigenome is also influenced by factors in the environment, our immune environment, by stochastic or random events that take place during development, and even by genetic variation itself. And so there are many ways by which two genetically identical individuals at birth could go through life and be exposed to different things and have different patterns of gene regulation that end up in the context of a difference in this case, health or disease. So one of the real interests in this field is obviously the way that the epigenome may act as an interface between our external environment and the way that genes are regulated and expressed in health and disease. And there's now increasing evidence that a whole range of environmental factors can have a direct effect on gene regulation via dysregulation of the epigenome. And I've put question marks there because this is, I think, a really new and exciting area of research, and we're really only in the infancy of understanding the extent to which these environmental factors can influence disease. What I would say here is obviously you can instantly see why this might also pose problems for understanding epigenetic patterns in the context of disease, and this is possibly going to be a feature of many of the talks that you hear today, and certainly through the process of the workshop, because many of these environmental exposures are obviously very confounded with the actual phenotypes that we want to look at, and so it's very difficult in many cases to delineate between epigenetic patterns that are associated with disease and those that are more directly caused by environmental exposures that might also be more prevalent in disease. Now, one of the kind of key findings from this work in environmental epigenomics is that there appear to be very critical periods of sensitivity. And the epigenome appears to be particularly vulnerable to external influences during the prenatal and very early perinatal period. And if you think about it, this makes sense. This is the time that those standard epigenetic signatures that define the different cell types are being laid down during development. There's lots and lots of DNA and cell replication occurring at that time. And because these epigenetic signatures, once established, can actually be maintained very stably across mitosis and down cell lineages, this provides a very nice mechanism by which exposures very early on in life, prenatally or post-natally, could have long-term consequences on health and disease. And obviously there's real growing interest really informed by some of the epidemiological findings that actually there are real developmental origins of many of the diseases that we're interested in studying, whereby the environment early on in life, particularly prenatally, may have effects on a disease like schizophrenia that maybe manifests itself later in adolescence or early adulthood. And as I said, so we know that there are critical windows of environmental sensitivity, certainly in the context of schizophrenia. This is just a figure from an article I wrote with Bart Rutten, almost 10 years ago now, actually, but this is still really a focus of a lot of work. The fact that we know there are environmental factors associated with schizophrenia, but the effects of those environmental factors appear to be specific to certain points in an individual's life course. And again, a real kind of focus of these environmental exposures appears to be this period of prenatal development. So coming to the work of our group, this slide just really summarises what our work is about and what we do. So we take an integrated genomics approach. Most of our kind of key work, I guess, is done on using post-mortem human brain tissue, and we're interested in a range of diseases: autism, schizophrenia, depression, dementia, bipolar disorder, but many others. Basically anything to do with the brain, we're interested in studying it. And we're also interested in characterizing the way that these epigenetic signatures and gene regulation changes across development and aging, and I'll talk in a minute about why understanding that is important. At the same time, we're kind of doing a lot of work in terms of model systems, both rodents. Joanna will talk about some of the zebrafish models, cell models, and like many others in the field, moving towards characterizing things at the level of single cell changes, particularly in the brain. A kind of key aspect of our work is putting together different layers of information. So we integrating genetic variation together with patterns of gene regulation and transcriptomics. And this takes some quite high-level, systems-level bioinformatic approaches. And I think that's kind of a real struggle for the field, is actually how do we put these data together in a very efficient way and ultimately understand more about the actual functional consequences of these changes. And just to really give you the kind of headline conclusion, I think, from what our work is telling us, is that these are all very heterogeneous disorders. So we know that no two individuals with schizophrenia or autism probably have the same underlying etiology. They may harbour different genetic variants, different levels of polygenic burden. They may be exposed to different environmental influences. But one kind of key message that we're finding is that at the molecular level, when we look at these readouts, transcriptomic or gene regulatory readouts from the tissues that are relevant for disease, we are gaining evidence for a very convergent molecular pathology. So here is just some data that Joanna generated. This is data looking at DNA methylation across the genome in prefrontal cortex tissue from many different brain banks, both in Europe and the US, finding differences at specific sites associated with schizophrenia. And I don't really want you to take away any details from this. This is all published. It's just to firstly say that we see many of these single site differences in DNA methylation associated with disease, and we see these across different regions of the brain, but also that individually each of these changes is actually relatively small in terms of its magnitude. And actually you need to really start thinking about the genome and gene regulation in a kind of systems level approach. And we know that genes don't obviously act in isolation. And a kind of nice way to think about this is thinking about transcriptional networks. And this is very much a kind of movement that the field is going into. And so a good analogy here about how biology works is to take these two kind of transport networks. This is focused on the northeast of the US. On the left is the road network, and you can see it's kind of very linear set of pathways interlinking the towns and cities. And on the right is the kind of airline network. And it's this pattern on the right which really is indicative, I think, of how biology works. And you could imagine if there was a huge snowstorm in a very minor town in say a huge snowstorm in Chicago, it would have a very limited effect in terms of the network shown on the left. But because Chicago is a key airline hub of that transport network, that will have a really dramatic effect in terms of the whole transport network across the whole of the US. And it's the same with the way that genes function in networks. So you can have a fairly small change in a very important gene that has huge downstream effects on the way that a whole network is functions and how it may relate to disease. And when we apply these kind of network approaches to our, in this case, DNA methylation data in schizophrenia, we firstly see that indeed gene regulation is organized in these very tight networks so that things co-vary together in very discrete ways. And you can, via some complex statistical analyses, actually put genes together into these networks. And then you can see how these networks themselves are differentially regulated in the brain tissue in schizophrenia. And here's just an example of two networks that we find differentially regulated in schizophrenia prefrontal cortex. What you'll notice here is that these are interlinked here by a gene called SHANK2. So this is a gene that's probably known to many of you. It's a key synaptic scaffold protein involved in synaptogenesis, brain development. It's been widely implicated in many central nervous system genes. The real interesting thing is that if you take the genes in these networks associated with schizophrenia, they're very highly enriched for pathways associated with neurodevelopment. So you probably can't read the text there, it's possibly a bit small, but these are all gene ontology pathways related to the development and functioning of the central nervous system. The other thing to note is that these the genes in these co-methylation networks are also highly enriched for the genes that have been implicated from the genetic GWAS studies of schizophrenia. There's a highly significant enrichment of these loci. And again, this goes back to my earlier point, so that if you remember that the genetic studies have been done on tens, maybe hundreds of thousands of individuals. And here, these study, epigenetic studies, because of the limitations of the tissue sources, are obviously done on much smaller numbers, maybe hundreds of individuals. But to me, it's really interesting that we're still converging on the same pathways, even with these much smaller sample sizes. That's telling us that, looking at the actual molecular pathology associated with these diseases in terms of gene regulatory patterns is really getting, I think, to the crux of some of the differences associated with disease. So we see kind of a real enrichment of neurodevelopmental genes in our schizophrenia brain, and the same is true of other neurodevelopmental disorders. This is unpublished work, the preprints on bioRxiv if anyone's interested. This is looking at autism brain. This is work by Chloe Wong, who was a PhD student in the group, has now got a PI position at the Institute of Psychiatry in London. And again, the take-home message here is firstly that you can really differentiate between cases and controls in terms of autism, non-autistic individuals based on their global DNA methylation profile. And this actually mirrors some of the other work we've done on these same brain tissue in collaboration with Dan Geschwind at UCLA and Shyam Prabhakar at the Genome Institute of Singapore, where we see very, again, convergent patterns of, in this case, histone H3K27 acetylation and also RNA-seq profiles in cases compared to controls. But the other take-home message from Chloe's work is that the differential methylation patterns are again enriched in genes that are involved in neurodevelopmental pathways. And of course, this is really interesting because we know that, it's certainly widely hypothesized that schizophrenia and autism and other neuropsychiatric disorders have a neurodevelopmental component in the context of their etiology. But one thing that's really lacking is actually detailed genomic data about human brain development. So most of what we know about the way that gene regulation changes across brain development is derived from rodent models. And while these have told us a lot, there are obviously very clear differences in the way that a mouse brain develops and functions across early life compared to a human brain. There are obviously differences in terms of gestational period. Much of a human brain development carries on developing after right through kind of adolescence, and there are clear temporal differences there. So it's important, I think, that we start to learn more about the actual patterns of gene regulation that take place during development of the human brain. And we have we're lucky to have a very good collaboration with a couple of brain banks in the UK that are focused on this prenatal period, the Human Developmental Brain Resource, and also the MLC Brain Bank network. It's a very unique set of samples that includes human fetal brain tissue going from about 23 days post-conception up to the end of second trimester, and we have about 180 of these samples now that we're profiling for different types of epigenetic modifications, DNA, different types of histone states, and also looking at transcriptomics and eventually moving into actually charting these changes at the single cell level. And so this is work again by another of my PhD students. This is Helen Spears. And what she did was profile DNA methylation across these samples. Again, I won't go into huge detail on the results. The paper's there published if you're interested. But again, the take-home message is that this is a real period of epigenetic plasticity. So you can see the very dramatic shifts in DNA methylation. This is just 4 example sites across the genome where you can see really strong linear changes in DNA methylation, the resolution of days across this period of brain development. And again, if you look at this in a kind of modular structure and actually look at the way that we see co-methylation modules changing across this period, again, you see that these changes are very coordinated. So we see kind of what we call modules of DNA methylation that kind of shift from a almost methylated to unmethylated status across this period. So plotted on the top of the module eigen genes of 2 of these modules, and you can just see the very dramatic shift. Each of these modules maybe contains several hundred loci across the genome, and these are genes that are being very dynamically shifted across this period. And one of the kind of really interesting things we found were that there were many sex differences in gene regulation during this period. So obviously you would expect these on the X chromosome or Y chromosome, but we see many sex differences in the way that gene regulation is occurring across locations on the autosome. So here's just examples of where males and females are showing very different patterns of DNA methylation right across this period of brain development. Perhaps more interesting are examples like this, where we actually see different trajectories, developmental trajectories in DNA methylation across development of the fetal cortex in males and females. So on the left is an example where males and females start off very different and converge towards birth, and on the right is an example where they kind of converge as development goes on. And of course this is really interesting given what I mentioned earlier about there being sex differences in the prevalence of many neurodevelopmental phenotypes, particularly autism. It firstly tells us that there are stable changes in gene regulation very early on in this period of brain development that's hypothesized to be important in these disorders, but also that these changes in many cases are transitory and may not be detected postnatally. And just to kind of follow up on that, this is a paper that's just been published. It's actually now published in Genome Biology. This is work in collaboration with Nick Bray in Cardiff, where we've taken the same samples and looked at gene expression using RNA-seq. And again, we find many examples of sex biases in fetal brain expression, even on many genes that are contained on the autosomes. And what's interesting here is that we see this at the specific transcript level. So specific isoforms are differentially expressed between males and females, and other isoforms of the same genes are less stably, less differentially expressed. The other thing we see is that male bias genes are actually enriched for expression in neural progenitor cells, whereas female bias genes are enriched in other types of cells, including reelin-producing cells and glia. And I think this is going to be really interesting for teasing apart some of the sex differences that we see in behaviour and also risk for neuropsychiatric disease. The other utility of these data is that we can start mapping regions of genes that we know are important in neurodevelopmental disorders, start mapping neurodevelopmentally important domains within those genes. And so here's just two examples. On the top is neurexin-1, which is obviously widely implicated in schizophrenia and autism. On the bottom is SHANK1, another gene that's often highly disrupted in autism brain. And plotted across the gene in blue are regions of the genome that which DNA methylation is highly dynamic across brain development. And what this tells us, what this enables us to do, is to focus our subsequent functional work, maybe in cells or other model systems, on these specific domains within the genes and start actually getting at the kind of functional importance of these genes during brain development and disease. And the other thing we can do is actually take the regions of the adult brain that we find differentially methylated in schizophrenia. So I apologise, it's a confusing chart, but each of these bars here represents a site in the genome that we find to be differentially methylated in the prefrontal cortex in individuals with schizophrenia. And on the y-axis for each of those sites is the extent to which that site is dynamic across human brain development. And there's actually a highly significant enrichment of these neurodevelopmentally dynamic loci amongst sites that are associated with schizophrenia in the adult prefrontal cortex. So again, this is further evidence that there's a neurodevelopmentally important component to the onset of schizophrenia. So here's just one example here. This is a site in the promoter of MYT1L. This is a very potent transcription factor that's involved in neurodevelopment. You can see the schizophrenia versus control difference in the box plot on the left. And on the right is just highlighting the extent to which this exact same site is highly dynamic across that period of human brain development. So these sites that we find differentially methylated in the brain appear to be the same sites that are playing a very key role in neurodevelopment. And it's not just DNA methylation that's dynamic across this period of the human life course. So this is data for hydroxymethylcytosine. Again, I'm not going to go into huge detail on this other than to say that this was once considered to be a mark that was A, not very prevalent, and B, not important in terms of differences and variation. Well, we actually find that this is a really dynamic modification, and that dynamicism goes in both ways, with sites becoming increasingly hydroxymethylated or becoming increasingly unhydroxymethylated across this period of brain development. And I think there's going to be real advances in understanding this modification as the technology for profiling it increases. And the other thing that we are now also studying, I've talked about our RNA-seq work, but we're also embracing some of the new sequencing technologies, some of the long-read sequencing technologies such as Pacific Biosciences and the Oxford Nanopore sequencing approaches that allow you to actually sequence right through entire transcripts. So one of the limitations of standard RNA-seq short reads is that it's very hard to actually define the different isoform structures of the RNA molecules that are being expressed. So using something like Pacific Biosciences, where we can read through the entire transcript, we can actually start characterizing and cataloging different isoforms that we see expressed in any given sample. And looking at isoform and splicing diversity is important because we know that actually most of our genes are expressed in these different forms, these different isoforms, and this is particularly prevalent in the central nervous system. It's something that's known to be developmentally dynamic, and it's important for understanding disease because we know that different isoforms can often have opposite or very different functional effects in many cases. And so differences in isoform abundance could be one thing that's maybe driving some of the pathogenic changes seen in gene regulation. And this is still very much work in progress. It's taken us a while to really get this technology working, and particularly in the context of the bioinformatics. It's a kind of whole different kettle of fish to mapping short read sequencing data. This is work led by Aaron Jeffries, a postdoc working with us in Exeter. And we've profiled now several fetal brain tissues, different regions of the brain, and also adult brain regions. This is just one example here. This is of a gene that's of interest in schizophrenia. It's one of the top GWAS nominated genes, TCF4, just to highlight the huge amount of isoform diversity that we see. In the top is fetal cortex, in the middle fetal hippocampus, and bottom is fetal striatum. Each of the black rows is a different isoform of that gene. And the take-home message here is that we find thousands of novel transcripts that are not in any current genomic annotation. So if you look in the UCSC genome browser or the GenCode database, these are isoforms that have not been described before. That many of the neuropsychiatric risk genes that we have been nominated from genetic studies are showing very considerable isoform diversity. And we're currently working to kind of map these different isoforms to some of the epigenetic modifications that I talked about. So I've talked about our work on neurodevelopment, but we're actually very interested in how